The task we're up to here is figuring out how life works.
At a very, very fundamental level, it's a key to understanding our species.
Everyone has a genome. We are all human, and it's the basis.
It is the word that means all of the DNA, all of the hereditary material of an organism.
The DNA is the script for life.
It's the stuff that tells one gene to make its machine in muscle cells and another gene to make its machine in brain cells.
In fact, that DNA code represents solutions evolved by life 3 billion years ago in some cases.
The human genome is essentially our shared inheritance. Bacteria have their genomes, and dogs have their genomes, and plants have their genomes, and we have our genomes.
Our DNA is almost identical with chimpanzee DNA. It's only about 2% different. Now, people probably aren't surprised that we share our genes with monkeys, but it turns out that we share about 50% of our genes with a tiny little roundworm, presumably a billion years ago or 500 million years ago. We had a common ancestor with the worms. And what we learn about those genes in worms and flies and yeast, now we can translate back to humans.
So we are just another species on this planet.
We are a very small species. The entire human race traces to a population of an effective size of only about 10,000 people in Africa, perhaps 150,000 years ago.
And because we're a very new species, we ain't been around that long, we actually share most of our DNA at a remarkably identical level.
We are all 99.9% identical. Of course, 0.1% difference out of a genome of 3 billion letters means any two of us also differ in 3 million separate places in our DNA.
Yeah, the bacteria won't come off right away. You might have to twirl a little. You might have to...
We all think of ourselves as completely unique individuals. And at some level, we are.
If you've seen identical twins, you've noticed that they look a lot alike. Well, they have exactly the same DNA between them. When you get to know them, you'll also find out that they're different in pretty significant ways.
But it's important for us to recognize how connected we are, how close we are with every other mammal, having the same sets of genes, the same body plan, how close we are with trees and funguses, which run their cells pretty much the same way we run our cells. The easiest way and most powerful way to see this grand unity of life is to read the DNA code. It's a 3 billion letter text.
If we printed out DNA sequence and put it in a book, you'd turn the pages, and what you would see would be a funny language, where there are only 4 letters in the alphabet, A, C, G, and T. Those are the 4 bases, as we call it, of DNA. It would take many of these books stacked on top of each other, about the height of the Washington Monument, in fact, in order to represent all of those 3 billion letters that you have inside each cell of your body.
And yet in that, those strings, in exactly the order of the letters, lies all of the fundamental secrets, lies all the instructions that our bodies use to build themselves from a single fertilized egg, the whole thing, the whole Taj Mahal of our bodies.
What we have when we want to analyze a genome is a huge problem.
Well, in the mid-1980s, a few people began to imagine that we might mount a carefully orchestrated attempt to read the entire sequence of the human genome.
The thought of sequencing the whole human genome was utterly outrageous when it was first proposed in 1985. People could barely sequence a couple of hundred letters of DNA at a time. It took a tremendous change in technology to make that possible.
you discover, that there are pessimists in this world, and, you spend your time with optimists.
What genomics is about is taking a global picture of all of the cell, all the DNA instructions, all the RNA readouts of the genes, all the proteins at once.
It's like, for example, mapping the continental United States. At the DNA sequence level, we certainly want building by building this whole map. We want yard by yard across the whole continent. We take the big picture, we divide it up, and we organise the smaller pictures relative to one another.
And again, congratulations to our colleagues in Germany and Japan.
The Human Genome Project is an international, multi-component effort to read out the instruction book for human biology, that is our genome, all of the DNA, and to understand what it's telling us.
We had to build all sorts of automated robotic systems to process hundreds of thousands of samples a day.
We work with about 1,000 plates of DNA here a day, or 100,000 DNA molecules sequenced here a day.
DNA starts at one end of the process in the lab and works its way through all the way down these hallways behind me as a large fragment of DNA.
The process of sequencing is determining the sequence of all the individual units in the DNA.
As it moves down the hallway, it gets chopped up into small bits, turned into many smaller bits of DNA that work their way through all of these robotic lines to get purified, sequenced.
It is a process that has to be repeated many, many, many times because of the size of the human genome.
And eventually hit a sequencer, which will give you the fluorescent readout of every base pair in that fragment of DNA.
Whilst we can get a sequence of bases in the genome, we don't know everything yet about how the bases are organised.
We're just beginning readers right now.
We have only the faintest glimmer of what a lot of this information is really telling us.
In a sense, we have the book of human life. We can read the letters, and now we're trying to find the words and put them together into sentences.
We are making this information completely available, automatically available nightly. It's just picked up and automatically sent to public databases.
This means that any scientist anywhere who has some idea can use this information to advance the question that they're working on. And this suddenly empowers all the brains of the planet.
So by putting it out daily, we're speeding discovery.
It's something about a big project where you've got a lot of people working, real synergism where the sum of the parts is more than the individual components.
It's giving biologists an opportunity to truly understand what's going on inside living systems and look at the parts that are actually moving inside, to understand how they are failing to work properly when they fail.
Deep breath. Good.
We wanted to have it to cure disease.
It's cloudy, but you do have some fluid up in here, so we were thinking...
We will now be in a position, fairly soon, to understand what underlies that otherwise mysterious and threatening reality, that some of us are strongly defended against certain threats and others are vulnerable.
Genes in general are there for very good reasons. We'd be in bad shape without them all working together to keep us healthy.
Just relax and breathe normally.
But if there's a glitch there, in a particular one, then it may not quite do its job, and then a disease may result. A certain set of illnesses, things like cystic fibrosis or sickle cell anemia or Huntington's disease, a particular kind of misspelling, almost certainly you're going to develop the disease. But many diseases aren't like that. The variation, the misspelling in the gene increases your risk, but it's not yes or no. A misspelling is simply a way of saying, oh, you got a G there instead of a T. or you had a GTCC there and now it's just GCC.
We will be able to predict those things to a very high degree once we are able to go in and say, and ask the question, which versions did you inherit, Albert, as against some other person?
Now, we all have these. There are no perfect specimens. We're all flawed at the DNA level. We have probably 30, 40, 50 glitches that are in places that we wish they weren't. So a lot of the focus is going to be on prevention. How do you keep people from getting sick in the first place?
Then, in time, therapies, medicines, etc., will come along, and it will be of great benefit to everyone.
I think it's very hard for most people to really grasp how fundamental and how revolutionary knowledge that's coming from genetic research is in terms of the decisions that they will be making in the future.
We're not talking about something that's distant from people. We're talking about things that are going to affect your reproduction, your health, your daily life.
You're going to get knowledge, and how do you deal with it?
When I get the chance, when I talk to people, I tell them that this information is coming and people have to know about it.
I think any kind of new knowledge is frightening until we understand it more.
We know that there have been bad experiences with people misinterpreting genetic information and using it for ill purposes in the past.
So we're engaged in a very interesting experiment. Is it in fact possible to study the ethical, legal, and social implications of a scientific revolution at the same time the scientific revolution is happening? We haven't, as a human race, had a particularly good track record there.
I think any project that has the capacity to affect so many lives, for good or for ill, has a fundamental obligation to study what is good and what is potentially wrong.
We can't be so afraid of it that we don't allow ourselves to use it to correct things that really cause undue human suffering.
What does it mean to understand the sequence of the human genome? Are we in danger of beginning to think of ourselves as marionettes that are manipulated by strands of DNA? And is this going to damage our view of ourselves as creatures of free will and spirit?
I think it should be obvious, and it isn't obvious, that we are far more than just our genes.
We're clearly a complex mixture of what our genes are telling us and what the environment does to us.
But I think as human beings, there are aspects of who we are that this sort of approach based on this chemical called DNA is probably not going to reveal the mysteries of. We won't understand a lot about human relationships. We won't understand what love means. We won't understand the aspects of us that are more spiritual than mechanical.
We've been trying to do science without even knowing the parts list, without even knowing the building blocks of a human.
But this is, I think, one of the most exciting moments in all of science, this adventure that we're on.
I'm sure 100 years from now, people will have better lives than they now do.
When I have an opportunity to sit back and think about the impact that this project is going to have on science in the future, I feel very excited and very proud to be part of it.
The rate at which we advance in trying to ameliorate human suffering will now go up extremely steeply.
And the kinds of moments that are comparable in my mind are big moments in history when Copernicus and Galileo discovered that the Earth was not the center of the universe. About 150 years ago, Darwin showed us our place in the animal kingdom. I think what's happening today is that we're finding out now at an internal level much more about what we really are.
This is humanity, this is Homo sapiens, this is it, this is the secret of our lives.